RNA synthesis and processing CSIRNet JRF Life Sciences. RNA synthesis and processing are central to cellular function, as they are the key steps in the flow of genetic information from DNA to protein. The process begins with transcription, where an RNA molecule is synthesized from a DNA template, and continues with various processing steps that convert the primary RNA transcript into a mature, functional RNA. The main types of RNA involved are messenger RNA, mRNA, ribosomal RNA, rRNA, and transfer RNA tRNA, all of which undergo specific processing steps before they perform their roles in protein synthesis. In this lecture, we will cover both RNA synthesis, transcription, and processing in detail, incorporating key points related to the CSIRNet JRF Life Sciences syllabus. 1. RNA synthesis, transcription. Transcription is the first step in gene expression, during which a particular segment of DNA is copied into RNA by RNA polymerase. The process involves several stages: initiation, elongation, and termination. Initiation. Transcription begins when RNA polymerase binds to a specific sequence of DNA called the promoter region. The promoter is recognized by the sigma factor in prokaryotes or transcription factors in eukaryotes, which guide RNA polymerase to the correct starting point. In eukaryotes, transcription begins with the binding of the transcription initiation complex, which includes transcription factors such as TFID, which binds to the TAA box, and RNA polymerase II. Elongation. During elongation, RNA polymerase moves along the DNA template strand in a 3' to 5' direction, synthesizing the RNA in the 5' to 3' direction. As the polymerase moves, the DNA template unwinds, and the newly synthesized RNA is elongated by the addition of ribonucleotides complementary to the DNA template. Termination. The termination of transcription occurs when RNA polymerase reaches a termination signal in the DNA. In prokaryotes, this can involve a terminator sequence that causes a You said, continue ChatGPT said. RNA polymerase to dissociate from the DNA, releasing the newly synthesized RNA transcript. In eukaryotes, termination is more complex and involves additional factors like the cleavage of the RNA transcript followed by polyadenylation, the addition of a poly A tail. In eukaryotes, transcription occurs in the nucleus, and there are three main types of RNA polymerases. RNA polymerase I synthesizes rRNA except 5' srRNA. RNA polymerase II synthesizes mRNA, snRNA, and some RNAs. RNA polymerase III synthesizes tRNA, 5' srRNA, and other small RNAs. Prokaryotes only have a single RNA polymerase responsible for the transcription of all types of RNA. The process is relatively simpler, but it still involves similar steps of initiation, elongation, and termination. 2. RNA processing in eukaryotes. After transcription in eukaryotic cells, the primary RNA transcript, also known as pre-mRNA, undergoes several modifications before it becomes a mature mRNA molecule capable of being translated into a protein. These processing steps include capping, splicing, and polyadenylation. Capping. Once the RNA transcript is synthesized, a 5' cap is added to the beginning of the mRNA. This cap is a modified guanine nucleotide, 7-methylguanosine, that protects the mRNA from degradation and aids in the initiation of translation. The 5' cap also facilitates mRNA export from the nucleus to the cytoplasm. Splicing. Eukaryotic genes often contain non-coding sequences called introns, which must be removed from the pre-mRNA. The remaining coding sequences, exons, are joined together in a process called splicing. This is carried out by a complex called the spliceosome, which recognizes specific sequences at the junctions of introns and exons. Alternative splicing allows a single gene to produce multiple mRNA variants, contributing to proteome diversity. Polyadenylation. After transcription is completed, a poly A tail is added to the 3' end of the mRNA. This tail consists of a long chain of adenine nucleotides, typically 200 to 250, and plays several important roles, including mRNA stability, protection from exonucleases, and facilitation of mRNA export from the nucleus. Polyadenylation is carried out by the polypolymerase enzyme after the mRNA cleaved at a specific site. 3. RNA editing and modifications. In addition to the primary steps of RNA synthesis and processing, there are other forms of RNA modifications and editing. For example, in RNA editing, specific nucleotides within the RNA sequence can be altered post-transcriptionally. One common example is adenosine to inosine editing, which occurs in certain mRNAs and can affect the coding potential of the RNA. 4. Differences between prokaryotic and eukaryotic RNA synthesis and processing. Prokaryotic transcription is relatively straightforward. There is no separation between transcription and translation. Translation can begin on the mRNA even while it is being transcribed. There are no introns in prokaryotic genes, so splicing is not necessary. Eukaryotic transcription is complex. Transcription occurs in the nucleus, and mRNA processing occurs before the mRNA is transported to the cytoplasm. Eukaryotic mRNAs are typically much larger, containing both exons and introns, requiring more extensive processing, capping, splicing, and polyadenylation. 5. Key regulatory elements in transcription and processing. Several regulatory elements control RNA synthesis and processing. Promoters and enhancers. These DNA sequences regulate the initiation of transcription. Promoters are located near the start site of transcription, while enhancers can be located further away and help enhance the transcription rate. Transcription factors. Proteins that bind to specific DNA sequences near the promoter to initiate transcription. In eukaryotes, the interaction of transcription factors and RNA polymerase II is crucial for accurate transcription initiation. RNA binding proteins. These proteins interact with nascent RNA molecules and participate in processes like splicing, transport, and stability. For example, SR proteins are involved in the splicing process, while HNRMPs, heterogeneous nuclear ribonucleoproteins, play a role in RNA processing and stability. Previous year questions. Question. CSIRNet 2021. Which of the following modifications occur in eukaryotic pre-mRNA during transcription? A. Methylation of the guanine base at the 5' end. B. Addition of a poly A tail at the 5' end. C. Splicing of exons. D. Deletion of exons. Answer. A. Methylation of the guanine base at the 5' end. This modification is a 5' cap, which is crucial for mRNA stability and translation initiation. Question. CSIRNet 2020. Which of the following RNA polymerases is responsible for the transcription of mRNA in eukaryotes? A. RNA polymerase I. B. RNA polymerase II. C. RNA polymerase III. D. All of the above. Answer. B. RNA polymerase II. RNA polymerase II is responsible for synthesizing mRNA in eukaryotes. Question. CSIRNet 2019. What is the function of the 5' cap added to eukaryotic mRNA? A. To protect the RNA from exonuclease degradation. B. To enhance the translation efficiency. C. To facilitate splicing of introns. D. Both A and B. Answer. D. Both A and B. The 5' cap protects the mRNA from exonuclease degradation and aids in translation efficiency by assisting ribosome binding. Conclusion. RNA synthesis and processing are fundamental processes that govern gene expression. In eukaryotes, transcription is followed by significant processing steps, including capping, splicing, and polyadenylation, all of which are essential for the production of functional mRNA. Understanding the molecular mechanisms involved in RNA synthesis and processing is critical for answering questions related to gene expression regulation, and this topic is frequently tested in the CSIRNet JRF Life Sciences exam. Make sure to review the key elements, such as the types of RNA polymerases, the function of the 5' cap, splicing mechanisms, and polyadenylation, as these are frequently assessed. Let me know if you'd like to proceed with another topic or need further clarification. You said. Transcription factors. ChatGPT said. Transcription factors. CSIRNet JRF Life Sciences. Transcription factors (TFs) are essential regulatory proteins that control the process of transcription by facilitating or inhibiting the binding of RNA polymerase to specific genes. They are key players in gene expression regulation, determining when and how much a gene is transcribed into RNA. Transcription factors function by recognizing specific DNA sequences called promoter regions or enhancer sequences in the genome, and they play a vital role in the activation or repression of gene expression in both prokaryotic and eukaryotic organisms. This lecture will provide an in-depth explanation of transcription factors, their types, mechanisms of action, and their role in regulating gene expression. The following aspects will be covered. Overview of transcription factors. Classification of transcription factors. Mechanisms of action. Cofactors and company regulators. Transcription factors in eukaryotes versus prokaryotes. Regulation of transcription factors. Previous year questions. 1. Overview of transcription factors. Transcription factors are proteins that bind to specific DNA sequences and are critical for the regulation of gene expression. They work by either recruiting RNA polymerase to the gene's promoter or by influencing the activity of RNA polymerase once it is bound to DNA. In eukaryotes, transcription factors typically function in conjunction with RNA polymerase II, which is responsible for transcribing mRNA. These transcription factors are part of larger complexes and often interact with other proteins to modify chromatin structure or facilitate transcriptional initiation. In prokaryotes, transcription factors interact with the RNA polymerase core enzyme to guide it to specific promoters, ensuring proper transcription initiation. 2. Classification of transcription factors. Transcription factors can be classified into two broad categories based on their function. General transcription factors (GTFs). These are required for the transcription of most genes and are involved in the formation of the transcription initiation complex. They help RNA polymerase bind to the promoter. GTFs include proteins such as TFID, TFIIA, TFIIB, TFIIF, TFIIE, and TFIIH, which are essential for the initiation of transcription by RNA polymerase II in eukaryotes. Regulatory transcription factors. These factors control the transcription of specific genes in response to various signals. They bind to enhancers, silencers, or promoters to regulate transcription rates. Some examples include activators, which enhance gene expression, and repressors, which inhibit gene expression. Regulatory transcription factors are often tissue-specific and may respond to signals such as hormones or environmental factors. Within regulatory transcription factors, there are various families based on their structural domains and DNA binding motifs, such as helix turn helix, zinc finger, leucine zipper, basic helix loop helix (BHLH). 3. Mechanisms of action. Transcription factors regulate gene expression through various mechanisms. Binding to promoter regions. Many transcription factors bind to specific sequences in the promoter region of genes, near the transcription start site. These sequences, such as the TAA box recognized by TFID in eukaryotes or the Pridnell box in prokaryotes, are essential for the initiation of transcription. Recruiting cofactors. Transcription factors do not always act alone. They often recruit cofactors such as coactivators, which enhance gene expression, or core repressors, which suppress transcription. Coactivators and core repressors help remodel chromatin structure, modify histones, or facilitate RNA polymerase's ability to transcribe DNA. Interacting with chromatin. Transcription factors can alter the structure of chromatin by interacting with chromatin remodeling complexes or histone-modifying enzymes, leading to either the opening or closing of chromatin regions. Open chromatin is more accessible for transcription, whereas condensed chromatin inhibits transcription. Modifying RNA polymerase activity. Some transcription factors can also regulate the activity of RNA polymerase itself, either by modifying the polymerase or interacting with elongation factors to enhance or inhibit transcription elongation. 4. Cofactors and company regulators. Cofactors and co-regulators are additional proteins that do not bind directly to DNA but assist transcription factors in regulating gene expression. They can be classified into two categories. Coactivators. These enhance the transcriptional activity of transcription factors. Examples include CBP-P300 and mediator complex, which help recruit the RNA polymerase complex and chromatin-modifying enzymes to the promoter. Core repressors. These inhibit transcription by interacting with transcription factors and preventing the formation of an active transcription initiation complex. NCOR and SMRT are examples of core repressors that recruit histone deacylases, HDACs, to compact the chromatin and inhibit transcription. 5. Transcription factors in eukaryotes versus prokaryotes. Eukaryotic transcription factors. In eukaryotes, transcription is more complex due to the presence of introns, exons, and chromatin. Transcription factors in eukaryotes require the formation of large complexes that interact with other regulatory proteins to initiate and regulate transcription. These factors also influence chromatin remodeling and RNA processing. Prokaryotic transcription factors. Prokaryotes have simpler transcriptional regulation. The RNA polymerase core enzyme, aided by sigma factors, binds to the promoter regions to initiate transcription. The sigma factors allow the polymerase to recognize specific promoters and direct transcription accordingly. In prokaryotes, transcription factors tend to act at the level of promoter recognition and help modulate transcription based on environmental cues. 6. Regulation of transcription factors. Transcription factors themselves are often regulated by a variety of mechanisms. Post-translational modifications. Transcription factors can be activated or inhibited by phosphorylation, acetylation, ubiquitination, or other modifications. For instance, the phosphorylation of transcription factors can influence their DNA binding ability or their interactions with cofactors. Signal transduction pathways. External signals such as hormones, growth factors, or stress conditions can activate or inhibit transcription factors. For example, the nuclear factor kappa B NF-kappa B pathway is activated in response to inflammatory signals, leading to the activation of transcription factors that regulate immune response genes. Feedback mechanisms. Transcription factors can be part of feedback loops. For instance, some transcription factors can regulate their own expression by binding to the promoter regions or interacting with other regulatory elements. 7. Previous year questions. Question. CSIRNET 2022. Which of the following transcription factors is involved in the initiation of transcription by RNA polymerase II in eukaryotes? A. TFID. B. TFIIF. C. TFIIH. D. All of the above. Answer. D. All of the above. TFIID binds to the TAA box. TFIIF helps stabilize RNA polymerase binding, and TFIIH has helicase activity and phosphorylates the RNA polymerase tail, all of which are required for transcription initiation. Question. CSIRNET 2021. The basic helix loop helix BHLH motif is a feature of which type of transcription factors? A. Repressors. B. Activators. C. Both. D. None. Answer. B. Activators. The BHLH motif is commonly found in transcription factors that act as activators, especially in the regulation of developmental processes and cell differentiation. Question. CSIRNET 2020. Which of the following statements is true about transcription factors? A. They only bind to enhancer regions in DNA. B. They regulate transcription by recruiting RNA polymerase to the promoter. C. They are always repressors. D. They are only involved in prokaryotic gene expression. Answer. B. They regulate transcription by recruiting RNA polymerase to the promoter. Transcription factors play a critical role in guiding RNA polymerase to the gene's promoter and modulating the transcription process. Conclusion. Transcription factors are fundamental in regulating gene expression in both prokaryotes and eukaryotes. Their ability to bind to specific DNA sequences, recruit cofactors, and interact with chromatin remodeling complexes ensures that genes are expressed at the right time and in the correct cell types. Understanding the various mechanisms by which transcription factors regulate gene expression is crucial for tackling questions in the CSIRNET JRF Life Sciences exam. Focus on key transcription factors, their families, and regulatory mechanisms to ensure a strong grasp of the topic. Let me know if you'd like to explore another topic. You said. Transcription factors machinery ChatGPT said. Transcription factor machinery CSIR Net JRF Life Sciences. Transcription is a highly regulated process that requires the coordinated action of multiple proteins and complexes. The machinery involved in transcription includes not only the transcription factors themselves but also a variety of additional components that enable the efficient and precise transcription of genes. This machinery facilitates the binding of RNA polymerase to the DNA template, the unwinding of the DNA helix, and the initiation, elongation, and termination of RNA synthesis. The role of transcription factors is central to this process, but they work together with other essential proteins and enzymes to ensure proper gene expression. This lecture will discuss the transcription factor machinery, focusing on the components involved in the initiation and regulation of transcription. The following points will be covered. Transcription machinery overview. RNA polymerase and its subunits. General transcription factors GTFs. Mediator complex. Chromatin remodeling complexes. Co-activators and company repressors. Post-translational modifications and regulation. Previous year questions. 1. Transcription machinery overview. The transcription machinery consists of a core set of proteins and complexes that work together to initiate and regulate the transcription of DNA into RNA. In eukaryotic cells, this machinery is especially complex due to the need to overcome chromatin compaction and the involvement of additional regulatory proteins. The key components of the transcription machinery include RNA polymerase II for mRNA synthesis in eukaryotes. General transcription factors GTFs. Chromatin remodeling complexes. Co-activators and co-repressors. Mediator complex. Together, these components ensure that transcription is not only initiated correctly but is also regulated in response to cellular signals, ensuring proper gene expression patterns. 2. RNA polymerase and its subunits. In eukaryotes, the transcription machinery is primarily driven by RNA polymerase II, which is responsible for transcribing protein coding genes into mRNA. RNA polymerase II itself is a large enzyme made up of multiple subunits. In its inactive state, RNA polymerase II requires the assistance of general transcription factors GTFs to initiate transcription. Key subunits of RNA polymerase II include RPB1, largest subunit. This subunit is responsible for the catalytic activity of the polymerase. RPB2, it helps form the catalytic core of the enzyme. RPB3 to RPB12, these subunits help with structural integrity and the proper functioning of the polymerase. Once the transcription machinery is assembled at the promoter region, RNA polymerase II begins RNA synthesis after the GTFs and other cofactors have set the stage for transcription initiation. 3. General transcription factors GTFs. General transcription factors GTFs are proteins required by RNA polymerase II to initiate transcription in eukaryotes. These factors are essential for the formation of the pre-initiation complex, PIC, which is a multi-protein assembly of the genes promoter that enables transcription initiation. The key GTFs include TFIID. This is the first GTF to bind the promoter. It is a complex of the TBP top binding protein and several TAS TBP-associated factors that recognize the top box and initiate the assembly of the pre-initiation complex. TFIIA. This factor stabilizes the binding of TFIID to the promoter and helps recruit additional GTFs. TFIIB. It recruits RNA polymerase II to the promoter and stabilizes the complex. TFIIB also helps in selecting the transcription start site. TFIIF. This factor helps stabilize RNA polymerase II binding to the promoter and facilitates the transition from initiation to elongation. TFIE. It recruits and activates TFIIH, which has helicase activity and phosphorylates the RNA polymerase II CTD C-terminal domain. TFIIH. This GTF is crucial for promoter opening and unwinding of the DNA to allow transcription to begin. It also phosphorylates the C-terminal domain of RNA polymerase II, facilitating the transition from initiation to elongation. 4. Mediator complex. The mediator complex is a large multiprotein complex that acts as an intermediary between transcription factors and RNA polymerase II. It serves as a bridge, transmitting signals from the regulatory transcription factors to RNA polymerase II. The mediator complex is essential for the activation of transcription and the recruitment of additional co-activators and chromatin remodeling factors. Mediator functions in several ways. It facilitates the interaction between activator proteins and the basal transcription machinery. It helps the transition from transcription initiation to elongation. It integrates signals from various transcription factors, ensuring coordinated gene expression. 5. Chromatin remodeling complexes. Since DNA is packaged into chromatin in eukaryotic cells, the transcription machinery must overcome the structural barriers posed by the chromatin. Chromatin remodeling complexes play a critical role in this process by modifying the structure of chromatin to make it more accessible to the transcription machinery. Some important chromatin remodeling complexes include SWISNF. These complexes use energy from ATP hydrolysis to alter nucleosome positioning, making DNA more accessible to RNA polymerase. ISWI. This complex is involved in nucleosome spacing and plays a role in chromatin remodeling during transcription initiation. NERD. This complex plays a dual role in gene repression and chromatin remodeling. 6. Co-activators and company repressors. In addition to the core transcription factors, co-activators and co-repressors play essential roles in fine-tuning transcriptional regulation. These proteins do not directly bind to DNA but work by interacting with transcription factors and modifying the chromatin environment of RNA polymerase activity. Co-activators. These proteins enhance transcriptional activity by facilitating the binding of RNA polymerase to the promoter or by promoting chromatin decondensation. CBP-P300 and mediator are examples of co-activators. Co-repressors. These inhibit transcription by recruiting histone deacylases, HDACs, or chromatin-modifying enzymes that compact chromatin and prevent transcription. NCOR and SMRT are examples of co-repressors. 7. Post-translational modifications and regulation. Transcription factors and components of the transcription machinery are often regulated by post-translational modifications, PTMs, such as phosphorylation, acetylation, methylation, ubiquitination, and simulation. These modifications alter the activity, stability, and interactions of transcription factors, providing an additional layer of regulation. For example, phosphorylation of RNA polymerase II at the CTD is a critical step in transcription initiation, elongation, and termination. Acetylation of histones opens up chromatin structure, allowing easier access for the transcription machinery. These modifications are dynamic and can be influenced by various signaling pathways, enabling cells to respond to environmental stimuli or developmental cues. 8. Previous year questions. Question. CSIR NET 2022. Which of the following general transcription factors is involved in the formation of the pre-initiation complex PIC in eukaryotic cells? A. TFIIH. B. TFIID. C. TFIIE. D. TVIV. Answer. B. TVID. TFIID is responsible for recognizing the top box and is the first factor to bind the promoter region to initiate the formation of the pre-initiation complex. Question. CSIR NET 2021. What is the primary function of the mediator complex in eukaryotic transcription? A. It binds directly to the top box. B. It bridges the interaction between regulatory transcription factors and RNA polymerase II. C. It helps with chromatin remodeling. D. It synthesizes mRNA. Answer. B. It bridges the interaction between regulatory transcription factors and RNA polymerase II. The mediator complex helps coordinate transcriptional activation by interacting with both regulatory proteins and the RNA polymerase complex. Question. CSIR NET 2020. Which of the following general transcription factors is responsible for helicase activity and DNA unwinding during transcription initiation? A. TP. B. TFIIH. C. TFIIF. D. TVIV. Answer. B. TFIIH. TFIIH possesses helicase activity and is essential for unwinding the DNA during transcription initiation. Conclusion. The transcription factor machinery plays a crucial role in regulating gene expression, ensuring that transcription occurs precisely and at the right time. By understanding the components of this machinery, including RNA polymerase II, GTFs, co-activators, and chromatin remodeling complexes, you can grasp the complexity of transcriptional regulation in eukaryotic cells. This knowledge is essential for answering questions in the CSI RNA JRF life sciences exam related to gene expression and regulation. Let me know if you would like further elaboration on any part of this topic. You said, formation of initiation complex, ChatGPT said. Formation of the initiation complex, CSI RNA JRF life sciences. The formation of the initiation complex is a critical step in the transcription process, enabling RNA polymerase to initiate transcription at the correct location on the DNA template. In eukaryotic cells, this process is complex and requires the assembly of a variety of proteins, including general transcription factors, GTFs, RNA polymerase II, and other cofactors. The initiation complex must be precisely formed to ensure that transcription occurs at the correct promoter and that RNA synthesis proceeds efficiently. This lecture will provide a detailed explanation of the formation of the initiation complex, with an emphasis on the various components and steps involved. The following points will be discussed. Overview of the initiation complex. General transcription factors, GTFs. Formation of the pre-initiation complex, PIC. Role of mediator complex and company activators. Chromatin remodeling. Phosphorylation of RNA polymerase II. Transition to elongation. Previous year questions. 1. Overview of the initiation complex. The initiation complex is a multi-protein complex assembled at the promoter region of a gene, allowing RNA polymerase II to initiate transcription. It includes the general transcription factors, GTFs, RNA polymerase II, and additional regulatory factors. This complex facilitates the unwinding of DNA, the formation of the transcription bubble, and the start of RNA synthesis. The steps involved in the formation of the initiation complex can be broken down into two main phases. Assembly of the pre-initiation complex, PIC. Activation of RNA polymerase II and transition to elongation. 2. General transcription factors, GTFs. The formation of the initiation complex in eukaryotes depends heavily on the general transcription factors, GTFs, which are required by RNA polymerase II for transcription initiation. These factors are recruited to the core promoter region of the gene, typically located upstream of the transcription start site. The main GTFs involved in the formation of the initiation complex include TFIID. This is the first transcription factor to bind to the DNA. The TBP TATA binding protein, subunit of TFIID binds to the TATA box, a conserved sequence found in many promoters. TFIID also includes several TAFs TBP-associated factors that help stabilize the binding of TBP to the promoter and further recruit other GTFs. TFIIA. This factor stabilizes the interaction between TFIID and the DNA. It helps prevent the dissociation of TFIID from the promoter and recruits other transcription factors. TFIIB. After TFIID binds to the promoter, TFIIB is recruited. It binds to both TFIID and RNA polymerase II, positioning the polymerase at the transcription start site. TFIIE. This factor recruits TFIIH, which is responsible for unwinding the DNA at the transcription start site, allowing RNA polymerase to access the DNA template. TFIIH. TFIIH has two key functions. It acts as a helicase to unwind the DNA and it phosphorylates the C-terminal domain (CTD) of RNA polymerase II, a critical step that triggers the transition from initiation to elongation. 3. Formation of the pre-initiation complex, PIC. The pre-initiation complex (PIC) is the assembly of RNA polymerase II and the general transcription factors. The process begins with the binding of TFIID to the TATA box in the promoter region. This is followed by the sequential recruitment of other GTFs. TFIID binds to the TATA box, forming the first stable protein DNA interaction. TFIIA binds to the complex, stabilizing TP's interaction with the DNA. TFIIB is recruited, which helps guide RNA polymerase II to the correct location at the promoter. RNA polymerase II then binds to the complex with the help of TFIIF. TFIIE and TFIIH are the last factors to be recruited. TFIIE helps recruit TFIIH, which has helicase activity, unwinding the DNA around the transcription start site. Once all these factors are assembled, the PIC is formed, and the RNA polymerase is positioned at the transcription start site. 4. Role of mediator complex and company activators. The mediator complex is a multi-protein complex that bridges the interaction between regulatory transcription factors, activators, and the core transcription machinery. While it is not directly involved in the assembly of the PIC, the mediator complex is crucial for the activation of transcription. It facilitates the interaction between enhancer-bound transcription factors and the pre-initiation complex. Additionally, co-activators like CBP P300 are recruited to the promoter region by transcription factors, assisting in chromatin remodeling and promoting transcriptional activation by modifying the chromatin structure. Co-activators can acetylate histones, leading to a more open chromatin configuration and facilitating access to the transcription machinery. 5. Chromatin remodeling. Chromatin structure must be modified to allow the transcription machinery access to the DNA template. Chromatin remodeling complexes such as SWI, SNF, and ISWI help move nucleosomes away from the transcription start site or modify histones to create a more open chromatin structure. These complexes use ATP hydrolysis to alter the positioning of nucleosomes, making the DNA more accessible to RNA polymerase II and GTFs. 6. Phosphorylation of RNA polymerase II. Once the PIC is assembled, TFIIH phosphorylates the C-terminal domain (CTD) of RNA polymerase II. This phosphorylation is a key regulatory step in the initiation process. The phosphorylation of the CTD enables the transition from transcription initiation to elongation. It also serves as a docking site for other factors involved in RNA processing, such as capping and splicing enzymes. 7. Transition to elongation. After the initiation complex is assembled and transcription begins, the RNA polymerase moves along the DNA template, synthesizing RNA in the 5' to 3' direction. As RNA polymerase II synthesizes the RNA, it is phosphorylated at specific residues, which helps transition from initiation to elongation. The phosphorylation of the CTD serves as a signal for the recruitment of additional elongation factors, and the RNA polymerase leaves the promoter region to proceed with RNA synthesis. 8. Previous year questions. Question. CSI RNET 2021. Which of the following transcription factors is directly responsible for the unwinding of DNA during transcription initiation? A. TFIIH. B. TFIIB. C. TFIID. DTP. Answer. A. TFIIH. TFIIH contains helicase activity, which unwinds the DNA during transcription initiation. Question. CSI RNET 2020. Which component of the transcription initiation complex is involved in the phosphorylation of RNA polymerase II, signaling the transition to elongation? A. TP. B. TFIIH. C. TP. D. Mediator complex. Answer. B. TFIIH. TFIIH phosphorylates the C-terminal domain (CTD) of RNA polymerase II, allowing the transition to elongation. Question. CSI RNET 2019. What is the primary function of the mediator complex in the initiation complex? A. It binds the TATA box. B. It bridges the interaction between transcription factors and RNA polymerase II. C. It is responsible for DNA unwinding. D. It phosphorylates the CTD of RNA polymerase II. Answer. B. Mediator complex. The mediator complex bridges the interaction between transcription factors and RNA polymerase II. Conclusion. The formation of the initiation complex is a highly coordinated process involving the assembly of RNA polymerase II, general transcription factors, chromatin remodeling complexes, and co-activators of the gene promoter. Understanding the roles of these components, as well as their interactions, is essential for understanding transcriptional regulation and the initiation of gene expression. This process is a fundamental concept in molecular biology and is often tested in the CSI RNet JRF life sciences exam. You said. Transcription activator ChatGPT said. Transcription activators CSI RNet JRF life sciences. Transcription activators are specialized regulatory proteins that bind to specific DNA sequences, often enhancers of upstream activating sequences, and significantly enhance the transcription of target genes. They are vital for the regulation of gene expression, acting as switches that determine whether a gene should be turned on or off, depending on cellular signals, environmental stimuli, or developmental stage. These proteins do not directly bind RNA polymerase but act through protein-protein interactions and chromatin modifications to facilitate the assembly and function of the transcriptional machinery. In eukaryotic systems, where chromatin structure poses an additional barrier to transcription, transcription activators are essential to making DNA accessible and recruiting the transcriptional machinery to the correct gene. They are typically modular in nature, with distinct domains responsible for DNA binding and activation. This lecture covers the following key areas: structure of transcription activators, DNA binding domains and activation domains, mechanism of action of activators, role in chromatin remodeling, interaction with coactivators and mediator, enhancers and activator binding sites. Examples of transcription activators. Previous year questions with answers. 1. Structure of transcription activators. Transcription activators generally consist of two functional domains. DNA binding domain (DBD). This domain recognizes and binds to specific DNA sequences, enhancers of upstream regulatory elements. Activation domain (AD). This domain interacts with coactivators, chromatin remodeling complexes, or general transcription factors to enhance transcription. Some activators may have additional domains for signal sensing, e.g., ligand binding domain and nuclear receptors dimerization, or regulatory control. 2. DNA binding domains and activation domains. Common DNA binding domains include helix turn helix HTH found in homeodomain proteins, zinc finger motifs seen in steroid hormone receptors, leucine zipper bzip found in proteins like CREB, helix loop helix VHLH found in transcription factors such as NIAID. Activation domains. Acidic activation domains found in yeast activators like Gal4 and BP16. Glutamine rich domains found in SP1. Proline rich domains found in CTF and F1. These domains do not directly contact the basal transcription machinery but work through protein-protein interactions. 3. Mechanism of action of activators. Transcription activators increase gene expression by several mechanisms. Recruitment of coactivators. They bind coactivators like CBP P300 which possess histone acetyltransferase activity, loosening chromatin. Interaction with the mediator complex. Activators help recruit the mediator, which bridges activators and RNA polymerase II. Stabilization of the pre-initiation complex picked by recruiting GTFs and RNA pol II to the promoter. Chromatin modification. Indirectly promote transcription by altering histone structure through recruitment of remodeling complexes. Activators may work synergistically, where two or more activators together lead to much higher transcription than each alone. 4. Role in chromatin remodeling. DNA is packed into chromatin, which can repress transcription. Activators recruit chromatin remodeling complexes like SWI SNF, which use ATP to reposition nucleosomes, making DNA accessible. They also recruit histone acetyltransferases HATs to acetylate histones and reduce their affinity for DNA, making the promoter region more open. 5. Interaction with coactivators and mediator. Coactivators do not bind DNA directly but are recruited by activators. Some key coactivators include CBP P300, histone acetyltransferases that modify chromatin to an open state. Mediator complex, a multi-subunit complex that physically links activators with RNA polymerase II and GTFs. Mediator acts as an integrator of signals and assists in the formation and stabilization of the transcription initiation complex. 6. Enhancers and activator binding sites. Activators bind to enhancers, which may be located upstream, downstream, or even within the genes they regulate. These enhancers work irrespective of orientation or distance due to DNA looping, allowing activators to interact with the promoter-bound transcription machinery. Enhancer promoter communication is facilitated by the looping of DNA, bringing distant regulatory elements in close proximity with the core transcription machinery. 7. Examples of transcription activators. Gal4 yeast recognizes UASG sequences, activates transcription via acidic activation domain. NF-kappa B activates immune and inflammatory genes upon stimulation. CREB, activated by phosphorylation via CAMP signaling, binds to CAMP response elements. P53, a tumor suppressor that activates genes involved in DNA repair, apoptosis, and cell cycle arrest. Estrogen receptor ER, activated by hormone binding, binds to estrogen response elements Aries. These transcription activators often respond to external signals and integrate intracellular pathways to fine-tune gene expression. 8. Previous year questions with answers. Question. CSIR-NET 2022. Which of the following is true regarding transcriptional activators in eukaryotes? A. They bind RNA polymerase II directly at the core promoter. B. They bind to top box in the promoter region. C. They bind to enhancer regions and facilitate transcription via coactivators. D. They unwind DNA at the transcription start site. Answer. C. They bind to enhancer regions and facilitate transcription via coactivators. Question. CSIR-NET 2020. Which of the following is a function of the acidic activation domain of a transcriptional activator like Gal4? A. DNA methylation. B. Direct RNA synthesis. C. Histone acetylation via recruitment of coactivators. D. DNA replication. Answer. C. Histone acetylation via recruitment of coactivators. Question. CSIR-NET 2019. Which of the following statements about transcription activators is incorrect? A. They bind to specific DNA sequences. B. They interact with the basal transcription machinery. C. They have histone methyltransferase activity. D. They often possess acidic or proline-rich activation domains. Answer. C. They have histone methyltransferase activity. Correct explanation. Transcription activators themselves do not have histone methyltransferase activity. They may recruit such enzymes indirectly. Conclusion. Transcription activators are key regulators of gene expression in eukaryotic cells. They perform their function by binding specific regulatory sequences, recruiting coactivators and chromatin remodeling complexes, and facilitating the assembly and activation of the transcription machinery. Their modular architecture allows flexibility and adaptability in gene regulation. Understanding their mechanisms, domains, and interactions is essential for cracking CSIR-NET JRF life sciences, as this area is frequently tested and holds significant conceptual weight. You said. Transcription repressor. ChatGPT said. Transcription repressors. CSIR-NET JRF life sciences. Detailed lecture. Maximum detail. Note table PYQs included. Transcription repressors are regulatory proteins that inhibit gene expression by interfering with the process of transcription. While transcription activators promote transcription by enhancing the recruitment and function of RNA polymerase and general transcription factors, repressors prevent transcription through multiple molecular strategies. These mechanisms are crucial for maintaining cell type specificity, temporal gene expression, developmental control, and response to environmental signals. Understanding transcription repressors is essential for the CSIR-NET JRF life sciences exam, especially given that questions often test knowledge of gene regulatory mechanisms, protein DNA interactions, chromatin structure modulation, and the function of specific repressor pathways. 1. Definition and importance. A transcription repressor is a DNA binding protein that negatively regulates gene expression by blocking the transcription of target genes. It binds to specific DNA sequences, often overlapping or adjacent to the promoter or enhancer regions, thereby preventing the access or function of the transcriptional machinery. This ensures that genes are expressed only when required, contributing to energy conservation and regulatory precision. 2. Types of transcription repressors. Repressors can be classified based on their mode of action. A. Competitive repressors. They bind to DNA sequences that overlap with or are adjacent to activator binding sites, thereby competing with activators for binding. This direct competition prevents the recruitment of coactivators or general transcription factors. B. Masking repressors. Quenching. They do not block DNA binding of activators but bind directly to the activation domains of activators, thereby preventing them from interacting with the transcriptional machinery or coactivators. C. Direct repression through core repressor recruitment. They recruit core repressor complexes such as histone deacetylases, HDACs, histone methyltransferases, or other chromatin modifying enzymes, which compact chromatin and make it inaccessible for transcription initiation. Deep repressor mediated chromatin condensation. They may actively recruit chromatin remodeling complexes that compact nucleosomes and silence gene expression, such as polycomb group proteins PCG. 3. Mechanisms of action: blocking RNA polymerase recruitment. Repressors combine to operate or silencer regions near the promoter to prevent the assembly of the pre-initiation complex, PIC, or block the recruitment of RNA polymerase II. Preventing enhancer promoter communication. By binding to insulator elements or silencing elements, repressors can prevent looping of DNA, thereby disrupting enhancer promoter communication required for transcription activation. Deep modulating chromatin state. Repressors recruit chromatin modifying enzymes. Histone deacetylases, HDACs, remove acyl groups from histones, causing tighter DNA histone interaction and transcriptional repression. Histone methyltransferases, HMTs, methylate histones to generate repressive marks like H3K93 or H3K273, which are associated with heterochromatin. Nucleosome remodeling complexes, compact chromatin or reposition nucleosomes to cover the transcription start site. 4. Silencer elements. Silencers are specific DNA sequences to which repressors bind. These elements can act at a distance and in an orientation-independent manner, much like enhancers. When bound by repressors, they inhibit transcription by preventing chromatin opening, PIC assembly, or enhancer function. 5. Examples of transcription repressors like REST and RSF, RE1 silencing transcription factor, represses neuronal genes in non-neuronal tissues by recruiting chorus and HDACs. Deep MADMAX complex. Opposes NYC-MAX activation by recruiting SYN3 core repressor and HDACs. Deep Groucho TLE family, in Drosophila invertebrates, recruited by repressor proteins to mediate gene silencing during development. ID, snail and slug, zinc finger repressors. Suppress E-cadherin expression during epithelial to mesenchymal transition, EMT, essential in development and cancer metastasis. Deep polycomb repressive complexes, PRC1 and PRC2, epigenetically silence genes through H3K27 methylation and chromatin compaction. 6. Transcriptional core repressors. Core repressors are non-DNA binding proteins that are recruited by repressors to execute repression. Examples include NCOR and SMRT, core repressors of nuclear hormone receptors that recruit HDACs. SYN3A, recruited by many repressors including MAD, REST, and MECP2. CTBP, C-terminal binding protein, interacts with many repressor proteins and brings HDACs and other chromatin modifiers. These multiprotein complexes often form large repressive assemblies on chromatin to silence transcription stably inheritably. 7. Biological significance. Developmental regulation ensures genes are turned off in inappropriate tissues, e.g., REST represses neuronal genes in non-neuronal tissues. Cell cycle control, repressors like RB protein block expression of S-phase genes. X chromosome inactivation, mediated by long non-coding RNAs and chromatin-associated repressor complexes. Cancer biology, mutations in repressors like p53 or deregulation of repressive chromatin marks are linked to tumorigenesis. 8. Previous year questions with answers 1. CSIR Net Life Sciences 2021. Which of the following is a true mechanism by which a transcriptional repressor can inhibit gene expression? A direct methylation of the promoter DNA via activation of histone acetyltransferase activity C binding to enhancer elements to facilitate activator function de-recruitment of histone deacetylases to the promoter region. Answer. De-recruitment of histone deacetylases to the promoter region. Explanation. This is a classical mechanism of repression wherein HDACs remove acyl groups, leading to chromatin condensation and transcriptional repression. 2. CSIR Net Life Sciences 2019. REST RE1 silencing transcription factor represses neuronal gene expression in non-neuronal cells by activating transcription factor CREBB recruiting coactivators to histone acetylate nucleosomes C binding to promoter and recruiting histone deacetylases D promoting enhancer promoter communication. Answer. C binding to promoter and recruiting histone deacetylases. Explanation. REST binds to RE1 sites and recruits chorus and HDACs to silence neuronal genes outside the nervous system. 3. CSIR Net Life Sciences 2020. Which of the following protein complexes is associated with transcriptional repression? A SWI-SNFB mediator CCBP P300. Deep polycomb repressive complex. Answer. Deep polycomb repressive complex. Explanation. PRC1 and PRC2 are known to repress gene expression by methylating H3K27 and compacting chromatin. Conclusion. Transcription repressors are indispensable to the precise control of gene expression, acting through DNA binding competition, masking activators, recruiting core repressors, and remodeling chromatin to a repressive state. Their roles span development, differentiation, metabolism, and disease, making them high priority targets in transcriptional regulation questions for CSIR Net JRF Life Sciences. A strong grasp of their mechanisms, examples, and differences from activators is crucial for conceptual and applied questions in the exam. You said RNA polymerases. ChatGPT said RNA polymerases. CSIR Net JRF Life Sciences. Detailed theory lecture. Maximum detail based on PYQ areas. No table includes PYQs with year and answers. RNA polymerases are essential enzymes responsible for the synthesis of RNA from a DNA template in a process known as transcription. These enzymes read the DNA template strand in the 3' to 5' direction and synthesize a complementary RNA strand in the 5' to 3' direction. In both prokaryotes and eukaryotes, RNA polymerases are central to gene expression, but their structure, number, and functions differ significantly between the two domains. Understanding RNA polymerases is crucial for the CSIR Net JRF Life Sciences exam because transcription is a fundamental process in molecular biology, and questions frequently test candidates on types of polymerases, their roles, inhibitors, subunit composition, promoter recognition, and associated factors. 1. RNA polymerase in prokaryotes. Prokaryotic organisms, such as Escherichia coli, have a single type of RNA polymerase responsible for transcribing all classes of RNA, mRNA, tRNA, and rRNA. Structure. Prokaryotic RNA polymerase is a multi-subunit enzyme. The core enzyme has 5 subunits. Alpha 2 copies involved in enzyme assembly and interaction with regulatory proteins. Beta, catalytic subunit responsible for RNA synthesis. Beta binds to the DNA template. Omega assists in enzyme assembly and stability. To initiate transcription, the sigma factor joins the core enzyme to form the holoenzyme, which can recognize and bind specific promoter sequences. Function of sigma factor. It directs the holoenzyme to the promoter by recognizing conserved sequences like the -10 prit box and -3 regions. After initiation, the sigma factor dissociates, and the core enzyme proceeds with elongation. Different sigma factors recognize different classes of genes under various conditions, providing regulatory specificity. 2. RNA polymerases in eukaryotes. Eukaryotic cells contain 3 primary nuclear RNA polymerases, each with distinct roles. A RNA polymerase I. coli, located in the nucleolus, transcribes a single gene type, the large rRNA precursor 45S, which is processed into 18S, 5.8S, and 28S rRNAs. B. RNA polymerase II, Pol II, located in the nucleoplasm, transcribes protein coding genes mRNA and also synthesizes snRNAs in some RNAs. Requires several general transcription factors GTFs for initiation, e.g., TFIID, TFIIH. Associated with CTD, C-terminal domain of the largest subunit RPB1, which undergoes phosphorylation to regulate initiation and elongation. C. RNA polymerase III, Pol III, located in the nucleoplasm. Transcribes ternus, 5srRNA, and other small RNAs. Each of these polymerases recognizes different promoter elements and utilizes distinct transcription factors to initiate transcription. 3. Mitochondrial and chloroplast RNA polymerases. These organelles possess bacteria-like single subunit RNA polymerases that are similar to T7 bacteriophage polymerase. They transcribe mitochondrial or chloroplast genes that are nuclear encoded but function within the organelle. 4. RNA polymerase structure and catalytic mechanism. RNA polymerases share conserved catalytic core characterized by a crab claw-like shape. The enzyme forms a transcription bubble, approximately 17 base pairs, by unwinding DNA. Nucleoside triphosphates (NTPs) are added one by one to the 3' of the Rome RNA chain, and pyrophosphate is released with each addition. The enzyme progresses with high processivity and fidelity, although proofreading is limited compared to DNA polymerases. The Mg squared ion is essential for catalysis, coordinating the phosphate groups of NTPs. 5. Inhibitors of RNA polymerases. Actinomycin D interpolates DNA and blocks elongation by all polymerases. Rifampicin specifically inhibits bacterial RNA polymerase by binding the beta subunit and preventing transcription initiation. Used against Mycobacterium tuberculosis. Alpha-omenidin. A toxin from Amanita phalloides mushroom. Inhibits eukaryotic RNA polymerase 2 at low concentrations and RNA polymerase 3 at higher doses. Folate is resistant. These inhibitors are often exam favorites in CSIRNet due to their specificity and practical applications in research and medicine. 6. Regulation of polymerase activity. RNA polymerase activity is modulated by transcription factors, chromatin structure, post-translational modifications, especially CTD phosphorylation in POL2, and interactions with enhancers silencers. The transition from initiation to elongation in POL2 is a highly regulated step involving kinases like TFIIH, which phosphorylates CTD, promoting promoter clearance. 7. Differences between RNA polymerase and DNA polymerase. RNA polymerase does not require a primer. It can initiate synthesis de novo. It lacks exonuclease proofreading activity, making RNA more error-prone. DNA polymerases require a template and primer, and they have higher fidelity with proofreading. Previous year questions with answers 1. CSIRNet Life Sciences 2018. Which one of the following toxins specifically inhibits eukaryotic RNA polymerase 2? A rifampicin B actinomycin D C-alpha-omenidin D-cyclohexamide. Answer: C-alpha-omenidin. Explanation: Alpha-omenidin inhibits RNA polymerase 2 at nanomolar concentrations, making it a potent tool in transcription studies and a common exam question. 2. CSIRNet Life Sciences 2020. In eukaryotic cells, which RNA polymerase is responsible for the transcription of ternal genes? A RNA polymerase IB, RNA polymerase 2, circ RNA polymerase 3, D mitochondrial RNA polymerase. Answer: cRNA polymerase 3. Explanation: RNA pol 3 transcribes ternus, 5srRNA, and small nuclear RNAs. 3. CSIRNet Life Sciences 2021. Which subunit of bacterial RNA polymerase recognizes the promoter region of DNA? A beta B beta, C alpha D sigma. Answer: D sigma. Explanation: the sigma factor is essential for promoter recognition and binding in prokaryotes. It is released after initiation. Conclusion: RNA polymerases are central to the gene expression machinery in all living organisms. Prokaryotes utilize a single enzyme with sigma factors for specificity, whereas eukaryotes possess 3 distinct polymerases, poli, 2, 3 each dedicated to transcribing specific types of genes. Understanding their structure, function, specificity, and inhibitors is critical for the CSIRNet JRF Life Sciences exam. Mastery of this topic ensures clarity in transcriptional regulation and builds a solid foundation for related topics like transcription factors, initiation complex formation, and RNA processing. You said, capping, ChatGPT said, capping. CSIRNet JRF Life Sciences, complete theory lecture, maximum detail based on previous year questions. No table includes PYQs with year and answers. Capping is a vital co-transcriptional modification that occurs at the 5' end of nascent free mRNA transcripts in eukaryotic cells. This modification results in the formation of a unique structure known as the 5' cap, which is essential for mRNA stability, nuclear export, efficient translation, and protection from exonucleases. The process of capping occurs almost immediately after the RNA polymerase 2 transcribes around 20 to 30 nucleotides of the mRNA. It is an indispensable quality control and regulatory step in eukaryotic gene expression and is not seen in prokaryotes. 1. Definition and structure of the 5' cap. The 5' cap is a modified guanine nucleotide attached to the first nucleotide of the RNA transcript via 5' to 5' triphosphate linkage, which is chemically unusual and resistant to nucleases. This cap is further methylated at the N7 position of the guanine base, resulting in 7-methylguanosine M to the power of 7G. In some cases, the ribose sugars of the first and second nucleotides are also methylated at the 2' position, forming cap 1 and cap 2 structures respectively. 2. Steps in the capping process. Capping involves 3 major enzymatic steps that occur co-transcriptionally. RNA triphosphatase activity. The enzyme removes the gamma phosphate from the 5' triphosphate end of the nascent RNA, leaving a diphosphate end. Gangliotransferase activity. This enzyme adds GMP, guanosine monophosphate, from GTP to the diphosphate end of the RNA through a 5' to 5' triphosphate linkage, creating the basic cap structure. Methyltransferase activity. The enzyme methylates the guanine at the N7 position, converting the cap to 7-methylguanosine M to the power of 7G, which is the hallmark of a functional 5' cap. Additional methylations may occur on the ribose sugars of the first and second nucleotides. 3. Enzyme complex and CTD involvement. The enzymes involved in capping are recruited by the phosphorylated C-terminal domain, CTD, of RNA polymerase 2. Specifically, the ester 5 phosphorylated CTD serves as a platform for assembling and positioning the capping machinery. This coordination ensures that capping is tightly coupled with transcription. 4. Biological functions of the 5' cap of protection from degradation. The 5' cap protects free mRNA from degradation by 5' exonucleases, increasing transcript stability. B. Facilitates nuclear export. Capping is required for the binding of cap binding complex, CBC, which mediates nuclear export of mRNA through the nuclear pore complex. C. Promotes translation initiation. In the cytoplasm, the cap is recognized by E40, a component of the eukaryotic initiation factor complex, which helps recruit the ribosome to the mRNA for cap-dependent translation initiation. D. Splicing efficiency. The presence of a cap enhances the efficiency of splicing of the first intron, indicating a role in coordinating multiple RNA processing steps. E. Quality control. Uncapped mRNAs are rapidly degraded, ensuring only properly processed mRNAs are translated. 5. Cap binding proteins. In the nucleus, the cap binding complex, CBC, binds the 5' cap and interacts with splicing and export machinery. In the cytoplasm, the 5' cap is recognized by E40, which is essential for ribosome recruitment and translation initiation. The decapping enzyme complex, DCP1, DCP2, removes the cap during regulated mRNA decay pathways like nonsense-mediated decay, NMD, and general mRNA turnover. 6. Cap-independent translation. Some viral RNAs and certain eukaryotic mRNAs initiate translation without a 5' cap, using internal ribosome entry sites iris. These are exceptions to the standard cap-dependent initiation. 7. Differences between capping in eukaryotes and prokaryotes. Capping is exclusive to eukaryotes. Prokaryotic mRNAs do not undergo capping. Instead, they possess a triphosphate at the 5' end and are rapidly translated, often co-transcriptionally, without the need for cap structures. Previous year questions with answers 1. CSIRNet Life Sciences 2017. Which of the following modifications occurs at the 5' end of a eukaryotic mRNA? Addition of poly A tail B removal of intron C addition of 7-methylguanosine demethylation of 3' end. Answer: C addition of 7-methylguanosine. Explanation: the hallmark of 5' capping is the addition of a 7-methylguanosine residue in a 5' to 5' linkage to protect and regulate the mRNA. 2. CSIRNet Life Sciences 2020. Which of the following statements about the 5' cap structure in eukaryotic mRNA is correct? It is added after transcription is complete. B. It promotes mRNA degradation. C. It is recognized by E40 during translation initiation. D. It is found in prokaryotic mRNAs. Answer: C. It is recognized by E40 during translation initiation. Explanation: the 5' cap is critical for translation initiation in eukaryotes, where E40 binds the cap and recruits the translation machinery. 3. CSIRNet Life Sciences 2019. The enzyme that adds a guanine residue to the 5' end of eukaryotic mRNA during capping is a poly A polymerase B gangliotransferase cRNA helicase spliceosome. Answer: B gangliotransferase. Explanation. This enzyme catalyzes the transfer of DMP to 5' and the RNA form in the Cas structure. Conclusion. Cavities is essentially a unique feature of eukaryotic RNA processing that plays a central role in the regulation of gene expression at multiple levels: transcription, splicing, export, translation, and stability. It represents one of the earliest quality control steps for RNA transcripts. Mastery of this topic is important for practicing CSI RNA life sciences, as questions frequently test both mechanistic understanding and functional consequences of RNA capping. Be sure to correlate this topic with RNA polymerase II, CTD phosphorylation, and translation initiation factors for integrated understanding. You said elongation. ChatGPT said elongation of transcription. CSI RNA JRF life sciences. Complete theory lecture. Maximum detail. Previous year questions included. No tables. Paragraph format. Transcription elongation is the central phase of gene transcription in which RNA polymerase travels along the DNA template, synthesizing complementary RNA strands by binding ribonucleotides in a 5' to 3' direction. Following initiation and promoter clearance, RNA polymerase transitions into a process of elongation complex. This phase is highly regulated and plays a critical role in ensuring transcriptional fidelity, efficiency, and responsiveness to various cellular signals. Transition from initiation to elongation. After the formation of the transcription initiation complex and synthesis of the first few nucleotides, typically around 10, RNA polymerase undergoes a conformational change, losing most general transcription factors in eukaryotes or sigma factor in prokaryotes. This allows it to escape the promoter and enter a stable elongation phase. In eukaryotes, this transition is highly dependent on phosphorylation of the RNA polymerase II C-terminal domain (CTD), particularly at SR2 residues, which allows recruitment of elongation factors and RNA processing machinery. 2. Mechanism of elongation. During elongation, RNA polymerase unwinds the DNA helix ahead of it to expose the template strand and rewinds it behind, forming a structure called the transcription bubble, typically 14 to 17 base pairs long. Within this bubble, about 8 to 9 nucleotides of the newly formed RNA remain base paired to the DNA template. The enzyme catalyzes the formation of phosphodiester bonds between the 3' group of the grown RNA chain and the incoming ribonucleoside triphosphates, RNTPs, which are selected based on Watson-Crick base pairing rules with the DNA template. 3. Elongation factors in eukaryotes. In eukaryotic cells, several elongation factors associate with RNA polymerase II to regulate elongation rate, pausing, and RNA processing. Key among them are TFIIS, resolves transcriptional pausing and assists in RNA cleavage to allow backtrack RNA polymerase to resume. DSIF, DRB, sensitivity inducing factor, and NELF, negative elongation factor, induce early elongation pausing near promoters. This is released by the action of PTFE, positive transcription elongation factor B, which phosphorylates RNA col 2 CTD, SR2, DSIF, and NELF. SPT5, SPT6, promote transcriptional elongation and chromatin reorganization. PTFE, CDK9 plus cyclin T, a major elongation activator, phosphorylates negative factors to allow elongation to proceed. 4. Pausing and proofreading. Transcription elongation is not always continuous. RNA polymerase can pause due to DNA sequences, chromatin structures, or incorporation errors. Pausing allows for co-transcriptional RNA processing like capping and serves as a regulatory checkpoint. Pause polymerase may either resume elongation or undergo backtracking, in which case the 3' end of the RNA is displaced from the active site. This requires transcript cleavage factors like TFIIS to realign the active site and allow elongation to resume. 5. Supercoiling and topoisomerases. As RNA polymerase progresses, it introduces positive supercoiling ahead and negative supercoiling behind the transcription bubble. This torsional stress must be resolved by topoisomerases, topo I and 2, which relieve supercoiling and facilitate smooth progression of elongation. 6. Chromatin and nucleosome dynamics. In eukaryotes, RNA polymerase must transcribe through nucleosomal DNA. This is achieved with the help of chromatin remodelers and elongation factors like FACT, facilitates chromatin transcription, which disassemble and reassemble nucleosomes. Histone modifications, such as acetylation, also make chromatin more accessible to polymerase during elongation. 7. Co-transcriptional RNA processing. As elongation proceeds, capping, splicing, and eventually polyanylation signals are added co-transcriptionally. The CTD of RNA polymerase II acts as a platform to recruit and coordinate these RNA processing machineries, making transcription elongation a hub of gene expression regulation. 8. Elongation in prokaryotes. In bacteria, the sigma factor is released after initiation, and core RNA polymerase continues elongation. Elongation factors like NUSA and GRIA assist in pausing and proofreading. Bacterial RNA polymerase also faces supercoiling challenges and pauses due to terminator sequences or regulatory proteins. 9. Transcription elongation and gene regulation. Recent studies show that elongation is a major regulatory point in gene expression. In metazoans, many genes are transcribed but held in a pause state until specific signals, e.g., stress or developmental cues, release RNA polymerase to complete transcription. This phenomenon is particularly common in developmentally regulated and inducible genes. Previous year questions with answers 1. CSI RNet Life Sciences 2016. What is the role of PTFE in transcription elongation? It inhibits elongation by binding RNA col 2. B enhances elongation by phosphorylating CTD and DSIFC cleaves backtrack RNA. D acetylates histones ahead of RNA col 2. Answer. B enhances elongation by phosphorylating CTD and DSIF. Explanation. PTFE phosphorylates SR2 of the CTD, as well as DSIF and NELF, converting paused RNA polymerase into a productive elongation complex. 2. CSI RNet Life Sciences 2018. Which of the following statements about eukaryotic transcription elongation is false? A TFIIS stimulates cleavage of nascent RNA to correct misincorporation via RNA polymerase II CTD is phosphorylated during elongation. C elongation is assisted by CAP complex where chromatin traversal de-elongation occurs after translation in eukaryotes. Answer. De-elongation occurs after translation in eukaryotes. Explanation. In eukaryotes, transcription and translation are spatially and temporally separated, and elongation occurs before translation in the nucleus, whereas translation occurs in the cytoplasm. 3. CSI RNet Life Sciences 2021. Which factor facilitates transcription elongation through nucleosomes? A Greb B fact circuit TFI-IDDU1SNRNP. Answer. B fact. Explanation. FACT facilitates chromatin transcription by temporarily displacing histone H2A, H2B dimers and reassembling nucleosomes after the passage of RNA polymerase II. Conclusion. Transcription elongation is not merely a passive extension of initiation, it is a highly dynamic, regulated, and coordinated phase of gene expression. It involves a complex interplay of elongation factors, chromatin remodelers, topoisomerases, and RNA processing proteins, all of which ensure accuracy, responsiveness, and proper maturation of transcripts. For CSI RNet JRF Life Sciences, understanding the molecular players and events in elongation is vital, especially in integrating it with gene regulation and co-transcriptional events like capping and splicing. You said. Termination. ChatGPT said. Termination of transcription. CSI RNet JRF Life Sciences. Complete theory lecture. Maximum detail based on previous year questions. No tables. Paragraph format. Termination of transcription is the final phase in the transcription process during which RNA polymerase ceases RNA synthesis, releases a newly synthesized RNA molecule, and dissociates from a DNA template. This step is crucial for defining the 3' end of the transcript and for recycling the polymerase for subsequent rounds of transcription. Termination mechanisms vary between prokaryotes and eukaryotes, and understanding these distinct mechanisms is fundamental to gene regulation. 1. Transcription termination in prokaryotes and bacteria. There are two main mechanisms of transcription termination. A Rho-independent intrinsic termination. Rho-independent termination is mediated purely by specific sequences within the DNA. The transcribed RNA contains a GC-rich palindromic sequence followed by a stretch of uracils. The GC-rich region forms a stable hairpin structure, stem loop, in the RNA, which causes RNA polymerase to pause. The U-rich sequence that follows forms a weak RNA-DNA hybrid due to AU base pairs, leading to destabilization of the transcription complex and release of the RNA transcript. B. Rho-dependent termination. This mechanism requires the Rho factor, a hexameric ATP-dependent RNA helicase. Rho binds to a Rut site, Rho utilization site, on the nascent RNA, which is AC-rich, G-poor, and unstructured region. It translocates along the RNA in a 5' to 3' direction using ATP hydrolysis. When RNA polymerase pauses at the termination site, Rho catches up and unwinds the RNA-DNA hybrid, causing the release of RNA and dissociation of the complex. 2. Transcription termination in eukaryotes. Eukaryotic transcription termination is more complex and polymerase specific. 2. Transcription termination in eukaryotes. Eukaryotic transcription termination is more complex and polymerase specific. A RNA polymerase termination. Termination of RNA coli transcription involves a termination factor called TTFI, transcription termination factor for coli, which binds to specific terminator sequences downstream of rRNA genes and induces pausing of the polymerase. Additional factors such as PTRF, polymerase I and transcript release factor, help dissociate the transcription machinery from the DNA. BRNA polymerase free termination. RNA col free terminates transcription at a short stretch of binding residues, T tracks, in the DNA template. Unlike pol A and pol 2, pol 3 does not require additional termination factors, and the RNA transcript is released immediately after the polymerase encounters the poly T sequence. cRNA polymerase 2 termination. Pol 2 termination is the most elaborate and involves coupling with pre-mRNA 3' end processing. There are two main models for pol 2 termination. Torpedo model. After transcription of the polyadenylation signal AAUAA, the pre-mRNA is cleaved by cleavage and polyadenylation factors such as CPSF and CSTF. The upstream RNA is polyadenylated and processed, while the downstream RNA, still attached to pol 2, is rapidly degraded by 5' to 3' exonuclease called XRN2 in humans. XRN2 catches up with pol 2 and induces its release from DNA. Allosteric conformational change model suggests that transcription of the polyadenylation signal causes a structural change in the RNA polymerase or the transcription complex, reducing its processivity and leading to dissociation. Most likely, both models act in combination, where a conformational change allows XRN2 to terminate the polymerase effectively. 3. Role of polyadenylation signals in termination. In pol 2 transcription, polyadenylation signals e.g., AAUAA and downstream GU-rich sequences are critical for both 3' end processing and transcription termination. The process involves recruitment of cleavage factors CPSF, CSTF, poly A polymerase, and other proteins. The coupling of cleavage and termination ensures that only full-length, properly processed transcripts are released. 4. Transcription termination and gene regulation. Termination is a regulatory checkpoint ensuring RNA molecules are full-length and ready for further processing. Defects in termination can lead to read-through transcription, transcriptional interference with downstream genes, and genomic instability. Moreover, alternative polyadenylation APA at different cleavage sites can result in mRNAs with varying 3' UTRs, affecting stability, localization, and translational efficiency, thus influencing gene expression regulation in development, differentiation, and disease. 5. Transcription termination in mitochondria. Mitochondrial transcription termination also involves unique factors like ENTR, mitochondrial transcription termination factor. These proteins bind to specific DNA sequences downstream of transcription units to pause or terminate transcription in a strand-specific manner. 6. Termination and recycling of RNA polymerase. After termination, RNA polymerase is recycled for another round of transcription. In bacteria, core RNA polymerase rebinds to sigma factor to initiate new transcription. In eukaryotes, dephosphorylation of the pol 2 CTD is necessary before it can be reused, which is carried out by CTD phosphatases like FCP1. Previous year questions with answers 1. CSIR Net Life Sciences 2015. Which of the following is required for rho-dependent termination in prokaryotes? AAGC rich hairpin in RNA poly T sequence and DNA seroprotein and red site depolyadenylation signal. Answer: seroprotein and red site. Explanation: rho-dependent termination requires the rho helicase to bind the red site on RNA and disrupt the RNA DNA hybrid using ATP hydrolysis. 2. CSIR Net Life Sciences 2017. Which of the following best describes the torpedo model of transcription termination in eukaryotes? A cleavage of RNA by RNase HB binding of rho factor to DNA. C5p to 3p exonuclease mediated degradation of RNA leading to polymerase release of RNA at poly T tract. Answer: C5p to 3p exonuclease mediated degradation of RNA leading to polymerase release. Explanation: in the torpedo model, XRN2 degrades the RNA downstream of the cleavage site and catches up to pol 2, triggering termination. 3. CSIR Net Life Sciences 2020. What is the role of the polyadenylation signal in RNA pol 2 termination? A pausing of ribosomes during translation facilitates 5' capping C recruitment of termination and cleavage factors D enhances mRNA nuclear export. Answer: C recruitment of termination and cleavage factors. Explanation: the polyadenylation signal is recognized by CPSF and CSTF, which mediate RNA cleavage and recruit polyadenylation machinery, also contributing to transcription termination. Conclusion: termination of transcription is not merely the end of RNA synthesis but an integral part of gene regulation and transcript maturation. Its diversity among different RNA polymerases and domains of life reflects its evolutionary importance. Understanding the molecular mechanisms behind both prokaryotic and eukaryotic termination, as well as their regulatory implications, is essential for mastering gene expression at the level required to excel in CSIR Net JRF Life Sciences. This knowledge also connects deeply with mRNA processing, stability, and the overall control of gene output in response to cellular demands. You said RNA processing. ChatGPT said RNA processing. CSIR Net JRF Life Sciences. Full-length theory lecture, paragraph format, no tables, maximum detail based on previous year questions. RNA processing is a crucial set of post-transcriptional modifications that precursor RNAs, pre-RNAs undergo to become functionally mature RNA molecules in both prokaryotic and eukaryotic cells, though it is more elaborate and regulated in eukaryotes. This includes capping, splicing, editing, polyadenylation, trimming, and chemical modifications, which collectively determine the stability, localization, and translatability of RNA transcripts. While prokaryotic mRNAs are usually translated directly after transcription with minimal processing, eukaryotic primary transcripts, especially pre-mRNAs, require extensive modifications before they can be transported to the cytoplasm and translated. 1. mRNA processing in eukaryotes. A5' capping. The addition of a 7-methylguanosine cap at the 5' end of nascent pre-mRNA is the first modification. The capping process involves the removal of the terminal phosphate by an RNA triphosphatase, addition of GMP by guanylyl and methylation of the guanine at the end 7 position by methyltransferase. This cap protects mRNA from exonucleases, facilitates nuclear export, and is recognized by cap binding proteins involved in splicing and translation initiation. B splicing. Pre-mRNA splicing is the removal of non-coding introns and joining of exons by the spliceosome, a large ribonucleoprotein complex consisting of small nuclear RNAs, SNR NOS U1, U2, U4, U5, U6, and associated proteins. The spliceosome assembles a conserved intronic sites, 5' splice site, branch point A, polypyrimidine tract, and 3' splice site, and catalyzes two transesterification reactions. Alternative splicing generates multiple mRNA isoforms from a single gene, increasing proteomic diversity. Splicing errors can lead to exon skipping or intron retention, often associated with diseases. C3p polyadenylation. After the cleavage of the pre-mRNA at a specific site downstream of the polyadenylation signal AAUAA, a poly A tail of approximately 200 adenines is added by poly A polymerase. This process is mediated by a complex of cleavage and polyadenylation factors including CPSF, CSTF, CFIM, CFIIM, and CAP. The poly A tail enhances mRNA stability, nuclear export, and translation efficiency by interacting with poly A binding proteins PABPs. 2. RNA editing. RNA editing is a post-transcriptional process that alters the nucleotide sequence of RNA, thus changing the coding potential of the transcript. It includes substitution editing, such as adenosine to inosine ATI editing by ADAR enzymes, and cytidine to uridine CW editing by APOBEC enzymes. Editing can alter codons, splice sites, or RNA secondary structure, thereby influencing gene expression and protein function. A classical example is the editing of apolipoprotein B, a POP mRNA, where a CW change introduces a premature stop codon, generating two isoforms, APOB100 and APOB48. 3. RNA splicing variants and regulation. Alternative splicing is a highly regulated process influenced by cis-acting elements, exonic, intronic splicing enhancers or silencers, and trans-acting factors SR proteins and HNRNPs. These interactions determine which exons are retained in the final mRNA. Regulation of splicing is crucial during development, cell differentiation, and stress responses. Misregulation is linked to diseases such as spinal muscular atrophy and cancer. 4. rRNA and Turner processing. Pre-rRNA transcripts 45S in humans undergo cleavage, base modification, methylation, and pseudoregulation, and assembly with ribosomal proteins to form mature 18S, 5.8S, and 28S rRNAs. These modifications are guided by small nucleolar RNAs, SNRNAs. Turner processing includes cleavage by RNase P, 5' end, and RNase C3, and addition of CCA sequence at the 3' terminus, and extensive base modifications e.g., methylation, thiolation, inosine formation critical for accurate decoding during translation. 5. mRNA surveillance and quality control. Eukaryotic cells possess RNA quality control mechanisms such as nonsense-mediated mRNA decay, NMD, non-stop decay, NSD, and no-go decay, NGD, to eliminate defective transcripts. NMD targets mRNAs with premature stop codons. NSD degrades mRNAs lacking stop codons. NGD clears transcripts with stalled ribosomes. These pathways ensure only functional RNAs are translated. 6. RNA transport and localization. Processed RNAs are selectively exported from the nucleus through the nuclear pore complex. mRNA export requires cap binding complex, CBC, exon junction complex, EJC, and TAP-NXF1 transport receptors. Specific RNAs are also localized within the cytoplasm for spatial regulation of translation, a process particularly important in neurons and embryonic development. 7. Small RNA processing, miRNAs and siRNAs. MicroRNAs, miRNAs, and small interfering RNAs, siRNAs, are processed from precursor transcripts by Drosha and Dicer, and then incorporated into RNA-induced silencing complex RISC. These small RNAs guide RISC to target complementary mRNAs to repress translation or induce degradation, playing crucial roles in post-transcriptional gene silencing and defense against viruses. 8. RNA processing in prokaryotes. Although less extensive, prokaryotes also process rRNA interna. A polycistronic precursor transcript is cleaved by RNases such as RNase 3 and RNase P to generate individual rRNAs internas. Ternas undergo further trimming and base modifications, though mRNA processing is minimal due to the absence of introns in nucleus. 9. Coupling of transcription and processing. In eukaryotes, RNA processing is tightly coupled to transcription by RNA polymerase II. The C-terminal domain CTD of RNA pol II acts as a scaffold to recruit processing factors during transcription. Phosphorylation of CTD regulates the sequential recruitment of capping enzymes, splicing factors, and polyadenylation complexes. This ensures precise and efficient RNA maturation. 10. Biological significance and disease association. Defective RNA processing is linked to several genetic diseases. For example, mutations in speciosomal components can lead to retinitis pigmentosa, and errors in polyadenylation have been associated with beta thalassemia. Aberrant editing and splicing patterns also contribute to cancer progression. Therefore, understanding RNA processing is vital not only for basic molecular biology but also for disease diagnostics and therapeutics. Previous year questions with answers 1. CSIR Net Life Sciences 2016. Which of the following statements is true regarding RNA splicing? A. It only occurs in prokaryotes. B. It removes exons from the transcript. C. It is mediated by the spliceosome in eukaryotes. D. It adds a poly A tail to mRNA. Answer. C. It is mediated by the spliceosome in eukaryotes. Explanation. The spliceosome removes introns and joins exons to form mature mRNA in eukaryotes. 2. CSIR Net Life Sciences 2019. A mutation that alters the AAUAA sequence in eukaryotic pre-mRNA would most likely affect a transcription elongation B splicing. C. Capping depolyadenylation. Answer. Depolyadenylation. Explanation. The AAUAA sequence is a canonical signal for 3' end cleavage and polyadenylation in eukaryotic mRNAs. 3. CSIR Net Life Sciences 2017. Which of the following RNAs is processed by both RNase P and tRNA nucleotide transferase? A. mRNA B terna C or RNA D mRNA. Answer. B terna. Explanation. RNase P processes the 5' end of tRNA, while tRNA nucleotide transferase adds the 3' CCA tail necessary for amino acid attachment. Conclusion. RNA processing is an indispensable aspect of gene expression regulation in eukaryotic cells. It ensures that RNA molecules are correctly modified, matured, and sorted for their respective cellular roles. Mastering the mechanisms and regulation of capping, splicing, editing, polyadenylation, and surveillance is essential for success in CSIR Net JRF Life Sciences. This knowledge connects core molecular biology to developmental biology, disease mechanisms, and biotechnology. You said, RNA editing. ChatGPT said, RNA editing. CSIR Net JRF Life Sciences. Comprehensive lecture, maximum detail, paragraph format, no tables, includes PYQs with answers in here. RNA editing is a post-transcriptional modification process by which nucleotide sequences of an RNA molecule are altered after transcription, leading to a final RNA sequence that differs from a genomic DNA template. It plays a vital role in expanding transcriptomic and proteomic diversity and fine-tuning gene expression in various organisms. Unlike RNA splicing, which removes introns, RNA editing changes specific nucleotides within the RNA molecule, thereby modifying its coding potential, stability, localization, or secondary structure. Types of RNA editing. The two major types of RNA editing in eukaryotes are substitution editing and insertion deletion editing. 1. Substitution editing. This is the most common form in higher eukaryotes, involving the chemical conversion of one base to another. Adenosine to inosine ATI editing. This type is catalyzed by a class of enzymes called ADARs. Adenosine deaminases acting on RNA. Inosine is interpreted as guanosine during translation and by the splicing machinery. This form of editing primarily occurs in the coding regions and untranslated regions (UTRs) of mRNA. ADI editing can create new splice sites, alter codons e.g., converting glutamine codon to arginine, and modify microRNA binding sites. Cytidine to uridine C to U editing. This occurs through APOBDC, apolipoprotein B mRNA editing catalytic polypeptide-like enzymes. A well-known example is the editing of apolipoprotein B mRNA, where a CAA codon is converted to UAA, a stop codon producing a truncated protein APOB48 in the intestine, compared to the full length APOB100 in the liver. 2. Insertion deletion editing. This is primarily observed in the mitochondria of kinetoplastic protozoans like trypanosoma. It involves the insertion or deletion of uridine U residues into the mRNA and is guided by guide RNase sternase. These sternase pair with pre-mRNA and direct the editing machinery to add or delete uridines at specific sites, thus generating a functional open reading frame. This type of editing can be extremely extensive and is essential for the expression of mitochondrial genes. Mechanism of RNA editing. ADI editing mechanism. The editing site is often within a double-stranded RNA structure formed by the exon or intronic complementary sequences. A DAR enzymes bind to these cs RNA regions and catalyze the hydrolytic deamination of adenosine to inosine. Since inosine pairs like guanosine, this leads to codon changes and affects protein structure and function. A DAR1 and a DAR2 are the two main isoforms involved in mammals. Editing specificity is determined by RNA secondary structure and sequence context. C-to-U editing mechanism. APOBDC1 binds to a morin sequence near the editing site and deaminates cytidine to uridine. Accessory proteins like ACF, APOBDC1 complementation factor, stabilize this complex and determine site specificity. Uridine insertion deletion mechanism in trypanosomes. The guanine binds to the pre-mRNA at the editing site and forms a mismatch. The mismatch guides the endonuclease to cleave the mRNA, followed by uridine insertion by terminal ribosyltransferase transferase 2-case or deletion by exonuclease. Finally, RNA ligase rejoins the edited RNA fragments. Multiple rounds of editing are often required for full transcript maturation. Biological functions and importance. RNA editing significantly increases transcriptome plasticity without altering the DNA sequence. In the nervous system, ADI editing affects ion channels, neurotransmitter receptors e.g., AMPA and serotonin receptors, and synaptic plasticity. Editing of Blur mRNA, for instance, changes the glutamine to an arginine, altering calcium permeability of AMPA receptors and protecting neurons from excitotoxicity. RNA editing also influences innate immunity, apoptosis, development, and circadian rhythms. Dysregulation of editing is linked to diseases like epilepsy, depression, glioblastoma, amyotrophic lateral sclerosis (ALS), and hepatocellular carcinoma. In plants, RNA editing is common in mitochondrial and chloroplast transcripts, mostly C to U changes. These edits restore codons for conserved amino acids and functional proteins essential for respiration and photosynthesis. Regulation of RNA editing. RNA editing is tightly regulated in a cell type specific, developmental stage specific, and stimulus responsive manner. Factors influencing editing include the availability and localization of editing enzymes, expression of GRNAs in kinetoplastids, RNA secondary structure, and interaction with RNA binding proteins. For instance, a DAR1 is induced by interferons and plays a role in modulating immune responses. Deficiency of a DAR1 results in unedited dsRNAs triggering MDA5 mediated innate immune activation. RNA editing versus RNA splicing. While both processes alter the mRNA post-transcriptionally, RNA splicing removes introns and joins exons to generate mature mRNA, whereas RNA editing changes the nucleotide content without cutting out RNA segments. Importantly, RNA editing can even alter splice sites and thereby indirectly influence splicing patterns. RNA editing in human health and disease. Alterations in RNA editing patterns are increasingly recognized as biomarkers and therapeutic targets. Reduced editing of Blur mRNA is associated with epilepsy and gliomas. Hyperediting can lead to overproduction of immunostimulatory dsRNA, promoting autoimmune diseases like Aicardi-Goutieres syndrome. Some cancers exhibit editing-dependent activation or silencing of tumor suppressor oncogenic transcripts. Thus, the RNA editing landscape serves as a critical layer of gene regulation, with potential for precision medicine. Previous year questions PYQs 1. CSIR Net Life Sciences 2015. In the intestinal cells, apolipoprotein B mRNA is edited to produce a shorter version of the protein. This is achieved by AAI editing by DAR-BC to U editing by APOBDC alternative splicing detranslational pausing. Answer. BC to U editing by APOBDC. Explanation. The C to U RNA editing by APOBDC1 converts CAA glutamine to UAA, stop codon truncating the protein into a POB48. 2. CSIR Net Life Sciences 2018. Which of the following is true about DAR enzymes? A. They convert cytosine to uracil B. They recognize single-stranded RNA C. They act only on tRNAs B. They convert adenosine to inosine. Answer. D. They convert adenosine to inosine. Explanation. A DARs are RNA-specific adenosine deaminases that act on double-stranded RNA regions and deaminate adenosine to inosine. 3. CSIR Net Life Sciences 2021. Insertion deletion type RNA editing is primarily seen in A human nuclear mRNA B is mitochondrial RNA C trichanosome mitochondrial RNA D bacterial mRNA. Answer. C trichanosome mitochondrial RNA. Explanation. In trichanosoma, RNA editing involves guide RNAs that direct uridine insertion or deletion in mitochondrial transcripts. Conclusion. RNA editing is a dynamic, sequence-specific, and enzymatically driven alteration of RNA nucleotides that broadens genetic information without modifying the genome. It represents a sophisticated post-transcriptional regulatory layer that is critical for normal cellular function and adaptive responses. Mastering the mechanisms, enzymes involved, and biological implications of RNA editing is essential to cracking the CSIRNet JRF life sciences exam, as it is a frequently tested and conceptually rich topic that integrates molecular biology, developmental biology, and disease biology. You said, splicing, ChatGPT said, RNA splicing. CSIRNet JRF life sciences, high yield, maximum detail lecture, paragraph format, no tables, PYQs with here and answer. RNA splicing is a post-transcriptional process in eukaryotes wherein non-coding introns are precisely removed from the pre-mRNA primary transcript and protein-coding exons are ligated to form a continuous mature mRNA. This process is a critical aspect of gene expression regulation and is essential for the generation of functional transcripts that can be translated into proteins. It occurs in the nucleus and is orchestrated by a large ribonucleoprotein complex known as the spliceosome. Constitutive and alternative splicing are two primary forms of RNA splicing. Constitutive splicing involves the removal of all introns and joining of exons in the order they appear. Alternative splicing allows for different combinations of exons to be joined together, producing multiple protein isoforms from a single gene. This dramatically increases the proteomic complexity without requiring additional genes and is particularly prevalent in higher eukaryotes, especially in neural and immune tissues. Spliceosome and its components. The spliceosome is a dynamic complex composed of 5 small nuclear ribonucleoproteins, SNRNPs, U1, U2, U4, U5, and U6, and numerous associated proteins. Each SNRNP contains a small nuclear RNA, SNRNA, that guides the splicing reaction through base pairing with specific sequences on the pre-mRNA. The splicing reaction is directed by consensus sequences found at the junctions of introns and exons. 5' splice site donor site typically begins with GU. 3' splice site acceptor site typically ends with AG. Branch point site, usually an adenine A located upstream of the 3' splice site within a conserved sequence. Polypyrimidine tract, rich in C and U nucleotides and found near the branch point. Mechanism of splicing, two-step transesterification reaction. Step 1, the 2' of the branch point attacks the phosphate at the 5' splice site, forming a lariat structure with a 2' minus 5' phosphodiester bond and releasing the 5' exon. Step 2, the free 3' of the 5' exon then attacks the 3' splice site phosphate, ligating the two exons together and releasing the intron in its lariat form. This reaction is catalyzed by the rearrangement and interactions of SNRNPs. U1 SNRNP recognizes the 5' splice site, U2 binds the branch point, and U4 U6 U5 tri-SNRNP is recruited to form the active spliceosome. After catalysis, the spliceosome disassembles and the SNRNPs are recycled. Alternative splicing patterns. Alternative splicing enables a single gene to produce multiple transcripts, and its types include exon skipping, mutually exclusive exons, alternative 5' or 3' splice sites, intron retention. This process is regulated by cis-acting elements, exonic, intronic splicing enhancers or silencers, and trans-acting factors like SR proteins, splicing enhancers, and HNRNPs, splicing repressors. Misregulation of these elements can cause diseases like cancer, spinal muscular atrophy, and myotonic dystrophy. Splicing and mRNA surveillance. The exon junction complex (EJC) is deposited near exon boundaries post-splicing. This complex plays roles in mRNA export, nonsense-mediated decay (NMD), and translation regulation. mRNAs with premature stop codons upstream of an EJC are targeted for degradation by NMD, ensuring faulty transcripts do not produce harmful truncated proteins. Self-splicing introns. Apart from spliceosomal splicing, self-splicing introns group 1 and group 2 exist primarily in lower eukaryotes, mitochondria, and chloroplasts. Group 1 introns utilize a free guanosine cofactor to initiate the splicing reaction. Group 2 introns follow a mechanism similar to spliceosomal splicing and are believed to be evolutionary precursors of the spliceosome. Clinical significance. Defects in splicing contribute to genetic disorders and oncogenesis. For example, mutations at splice sites can lead to exon skipping or intron retention. Mutations in SMN1 gene splicing lead to spinal muscular atrophy. Many cancers show altered splicing of tumor suppressors and oncogenes, e.g., BCLX long versus short isoforms. Antisense oligonucleotide therapy, such as Spinraza for spinal muscular atrophy, works by modifying splicing patterns to restore functional protein production. Previous year questions PYQs 1. CSIRNet Life Sciences 2016. The lariat structure formed during splicing contains a unique phosphodiester bond. It is A 2' minus 5' between branch point A and G at 5' splice site B 3' minus 5' between branch point A and G C 2' minus 5' between branch point G and at 3' and D 3' minus 5' between branch point A and U. Answer. A 2' minus 5' between branch point A and G at 5' splice site explanation. The 2' of the branch point attacks the 5' splice site G, forming a 2' minus 5' phosphodiester bond. 2. CSIRNet Life Sciences 2018. The recognition of the 5' splice site in pre-mRNA is primarily done by AU2 SNRNP BU4 SNRNP CU1 SNRNP DU5 SNRNP. Answer. CU1 SNRNP. Explanation. U1 SNRNP base pairs with the 5' splice site of the intron during the early stages of spliceosome assembly. 3. CSIRNet Life Sciences 2022 alternative splicing increases. A the number of genes in the genome, B the number of terners. C the diversity of proteins without increasing gene number, D the replication rate of DNA. Answer. C the diversity of proteins without increasing gene number. Explanation. Alternative splicing allows one gene to encode multiple protein isoforms, enhancing proteomic diversity. Conclusion. RNA splicing is a vital mechanism in eukaryotic gene expression that ensures the accurate and flexible processing of pre-mRNA into mature, translatable mRNA. The ability to selectively include or exclude exons through alternative splicing enables functional diversification of proteins. Understanding the molecular components and mechanisms of the spliceosome, along with its role in health and disease, is crucial for mastering this topic in the CSIRNet JRF Life Sciences syllabus. This topic is repeatedly tested in conceptual and clinical formats, making it indispensable for exam success. You said, polyadenylation. ChatGPT said, polyadenylation. CSIRNet JRF Life Sciences, maximum detail, paragraph format, no tables, PYQs included with here and answers. Polyadenylation is a post-transcriptional RNA processing event in eukaryotes wherein a stretch of adenine nucleotides, called the poly A tail, is added to the 3' end of the nascent pre-mRNA. This event is critical for the stability, nuclear export, and translation of mRNA. The poly A tail also plays essential roles in mRNA surveillance and regulation of gene expression. It occurs co-transcriptionally, usually after the transcription of the gene is complete, and is tightly regulated by multiple protein complexes. Signal for polyadenylation. Polyadenylation begins with the recognition of a highly conserved hexanucleotide sequence motif, AAUAA, located about 10 to 30 nucleotides upstream of the cleavage site in the 3' untranslated region, 3' UTR of the mRNA. A GU-rich or U-rich downstream sequence element, DSE, is often found 20 to 40 nucleotides downstream of the cleavage site and is essential for the efficiency of cleavage and polyadenylation. These sequence motifs are recognized by a set of protein complexes including CPSF cleavage and polyadenylation specificity factor binds the AAUAA signal. CSTF cleavage stimulation factor binds the GU-rich DSE. CFI and CFII cleavage factors I and 2 aid in the cleavage.